introduction, and thanks very much for the invitation to speak with you all this evening. It's a real pleasure to be here. I am not going to talk about actual paintbrushes that you paint things with. Instead, I'm going to talk about a group of wildflowers that are commonly called the paintbrushes. And here I have a really nice picture of some paintbrushes from British Columbia. We'll see this picture a little bit later. But before I get into kind of the details about the group itself, I wanted to set the stage a little bit with kind of a brief introduction to what my research is about, what I do, and the type of data that I collect. So I really like to open up by kind of pretending that I'm introducing myself for the first time to someone. And usually I say I am a botanist, and then depending on their reaction to that phrase, I might give them more detail. And when I do give them more detail, I tell them that I'm a plant systematist and an evolutionary biologist. And it's a pretty big mouthful of words to describe what I do, but this figure here on the left side of the screen really nicely illustrates the kind of core components of my work as a systematist. And you'll see that it incorporates a lot of components of evolution as I walk through it. So this is a figure by Todd Stussy from a late 70s paper. And the three main parts of a systematist, as he kind of outlines, are composed of taxonomy, phylogeny, and evolution. Taxonomy is very kind of closely tied with classification. And this is the process where we describe species and we organize and classify them into a way that allows us to extract information from the classification and to insert information into that system. The study of phylogeny is basically trying to understand the evolutionary relationships among organisms. A really kind of nice way to think about this is figuring out the family tree of all of the organisms that live on Earth. So that's the study of phylogeny. And then another core part of a systematist's work is studying the process of evolution. And these are processes that generate the diversity that we name during taxonomy and that we study when we build phylogenies. And these three components really kind of talk to one another, they inform one another, and they feed back on one another. So for example, taxonomy is where we start when we want to build a new phylogeny. Building phylogenies incorporates information that we know about the process of evolution. And phylogenies and taxonomies help us to inform the process of evolution in itself. So each of these pieces of the pie really feeds in, and it's kind of this dynamic thing that's very exciting. So systematics. But really, I think that taxonomy and classification are kind of the core of this work. And it's certainly what got me interested in science in the first place. So classification is something that we do really naturally and very easily. And I can guarantee that each one of you has participated in classification to some extent. So for example, if you're hiking, walking in the woods, as you're walking down this road, you're probably very naturally beginning to classify the variation that you see in the world around you. You can very quickly discern things like the grasses from the shrubs from the trees. And as you begin to look closer, you can find even more ways to classify the things that you see around you. So for example, if we kind of narrowed our focus to just the trees that are in this picture, and we begin to look closer at them, we would realize that there's actually a diversity of trees here, and they represent another kind of level in our classification. And we would begin to notice things about them like, their cones look different, and their leaves look different, and their habits look different. And so when we put organisms in little bins and boxes in species, we're participating in classification and participating in taxonomy. This can be a very easy thing to do. when we're kind of confronted with a collection of species that looks something like this, distinguishing them is super easy, right? Like all of these things look very different from one another, and we can see that each of them are distinct and unique entities. But sometimes distinguishing species can be really, really difficult. And this is a great example of that. So here we have a collection of beetles that, to my unknowledgeable eye, all look very similar to one another. And so if you asked me what the question or how many species there are in this picture, it would be really hard for me to say something other than one species. And it's often in times of like this, where things are morphologically, like the way they look is very, very similar, that we start to lean on other types of data to help us understand where species boundaries are, where different species exist. So one of those other sources of data that I use is, again, this idea of phylogenies, that I introduced in the first slide. So again, phylogenies are a way that we depict evolutionary relationships among organisms. And really, these are just diagrams that help us to organize these relationships in a way that we can read relatively quickly. So this is kind of a mock phylogeny. I made this one up completely. And it's composed of three kind of important parts. There are, well, I'll start by saying the way that we read these is kind of with time. And so time is going from the left side of the screen to the right side of the screen. So distantly in time is on the left and the present is on the right. So all of those horizontal lines are what we call the tips of the tree. They represent what's happening at the present in this particular moment. And they're connected by a series of horizontal lines called branches. And those branches come together at things called nodes. And the nodes represent past relatives, ancient relatives that connect currently existing species to one another. And so we read these along through time, and we don't have to use these phylogenies as our diagram. We can use kind of more simplified versions. So for example, we might think about the process of speciation and how new species are formed as being something that looks like this. So we have some population in time that's represented by this circle. As time goes by, populations kind of separate and they begin to distinguish themselves from one another. This could be branches on that tree, until at some point in time, things are very different from one another. And so these could be the different tips on the tree. So this is just kind of another way to think about how we read these phylogenies. So when I use phylogenies to study species, I am oftentimes making comparisons between one part of the tree and another part of the tree. So here I'm looking at tips from the very top and those from closer to the bottom. And you can see where that blue point is. That's the node where they have a common ancestor. And that is relatively long ago in time. And so usually we hypothesize that these kinds of comparisons are made across very old lineages or old divergences. These are diverged populations. But sometimes I'm making comparisons more recent in time, in shallower parts of the tree, where populations are in the early stages of speciation and they're beginning to diverge from one another. So this is kind of that framework where I'm trying to understand how speciation happens and where species are distinguishable from one another. But in both of these cases, in order to kind of apply this framework, I really need to know what species are in the first place. I need to be able to distinguish populations that have diverged, that are kind of good species, and those that have not diverged yet. They're in the early stages. So, when it is challenging to define species from the way that they look, I will oftentimes look at the phylogeny and try to understand if they are in the early stages of diversification, what we call incipient species. These are usually really young lineages and they have those shallow nodes that connect them. But sometimes I'm making comparisons across diverged populations in places where we would have otherwise thought that they were good species. And these are particularly confusing cases because usually they look morphologically very similar, but their genome tells me, their evolutionary history tells me that they're actually quite distantly related. And one of the things that happens naturally in populations that kind of confuses the signal is something called hybridization, which we encounter a lot in plants. And it really makes our challenge of defining species much more difficult. So, what I've been talking about up to now is really thinking about species as being distinct entities or not. They have diverged or they haven't. But the truth is that speciation is a very continuous process. It's always happening. It never stops. And so when we define species, it's very much at a snapshot in time. And so many evolutionary biologists are really trying to push away from an either or kind of comparison and talk about this as more of a continuous process. And so when I frame my work now, I really tend to think of it more as a process of speciation and just trying to identify where species are in this process. So this is a really simple diagram, very similar to a phylogeny, that's trying to do that exact same thing. Here, I like to imagine that there's a population at the bottom of the diagram that is composed of lots of individuals that look very similar. And as time goes by, these populations begin to acquire differences that distinguish them. They can be things like morphology, the way that these populations look, genomic data, like what their genome looks like, or ecological or habitat associated differences. But anyway, as these differences are acquired, as more and more time goes by, these populations become so distinct from one another that that they are very clear and distinct. But ultimately, there's speciation still continuing in each of these places too. So it's a continuous process. So my job as a systematist is to try to understand where lineages are in this process to help us understand where species boundaries are. And then in the context of studying the process of evolution and trying to understand something about evolution, the outstanding questions that I ask in my research are, what are the criteria that are acquired by species as they are formed in those early stages? And is there some sort of order that these criteria are acquired in? Do we see differences in the way they look first? Or do we see differences in their habitat first? All of this information will go in to help us understand how the process of evolution proceeds. Okay, so that's all of the theory for right now. This hopefully kind of sets the stage for how I think about species and how I think about this really charismatic group of plants that I'll spend the rest of the time talking about. So again, I work with the genus Castilleja. These are more commonly known as the paintbrushes. And if you've ever seen paintbrushes before, this might look familiar to you. However, this is actually a collection of wildflowers here. The paintbrushes are these bright pink, pretty ones kind of in the foreground. This was a picture that was taken in British Columbia. So this species doesn't actually exist in our neck of the woods, but many of our local Castellaius look a lot like this. But they're really beautiful wildflowers, and they're oftentimes kind of associated with other flowers that have strikingly different colorations, and they're just extremely photogenic. And really, it's kind of hard to live in Western North America and never have seen something that looks like this. So, even if you haven't been to the mountains and seen, the beautiful, the green hillsides with these bright red wildflowers, these are Castilleja. Even if you haven't seen this, you've probably seen it on a calendar or a postcard or a mug, a keychain. Like, Castellais is literally everywhere, and for good reason. It's one of the most kind of iconic wildflowers in North America, and particularly Western North America. They're just beautiful. But Castilleja aren't all big and red and showy like we see in these pictures. There's actually a lot of diversity in Castilleja, and this picture kind of illustrates some of that diversity. So here I've got a collection of different shapes and sizes and colors, and they really kind of run the gamut in terms of how showy they are and how big they are. So, you know, one thing that Castilleja has is a bunch of notoriety about how challenging it is taxonomically. So I love to show this quote. This is from Harold Rickett, who wrote this really great kind of collection of books from called The Wildflowers of North America. And he wrote them in different sections. And so this quote comes from a portion of that publication that's specific to the Northeast, where we don't have very many species of Castilleja. Anyway, he writes, "The genus Castilleja is one of those that make botanists wish they had embraced some easy branch of science, such as theoretical physics. It is a Western genus with at least 250 species, on the exact characteristics of which no two botanists seem able to agree. Fortunately for the readers of the present volume, only 4 species are found in the northeastern states, and they are not hard to distinguish." So, you know, this is a funny quote. You know, he's saying like taxonomy in Castilleja is bonkers. It's like super hard. They're really challenging. Castilleja just, it's so hard. I'd rather do physics. And he's not wrong. This is a really challenging group of plants. And so I think a logical kind of next question is like, why? Why is this group so taxonomically difficult? What genus are really hard to distinguish from one another? And one of the reasons why they're hard to distinguish from one another is because the diagnostic traits, you know, the traits that we use to decide where species are relatively continuous across species boundaries. And so when he says that at least 250 species in the genus on the exact characteristics of which no two botanists seem able to agree, he's absolutely right. Most botanists will argue actually about where species are in Castilleja, what is a species, what isn't a species, even what the identity of an individual is. So these diagnostic traits that we typically use to identify species have to do with a couple of different things. So the ecology and the geography of where an individual or a species occurs. So this is basically where the plant is and what kind of habitat it lives in. We also use traits associated with their morphology. So what the plant or the species looks like. This also gives us a lot of insight into the biology of the plant itself. And then kind of in modern day systematics, we also use information from the genome. So these phylogenies that I introduced you to. And these are genetic characteristics that help us to understand how species are related to one another and how speciation has happened in the group. So in the next set of slides, what I want to do is kind of walk you through some of the variation that we see in Castilleja to kind of illustrate the enormous amount of variation in the group, but also to try to give you some insight into how challenging the taxonomy can be. So I'm going to start by talking about the diagnostic traits associated with ecology and geography. So Castilleja occurs in a very broad geographic context. So it's found in North America and in South America. And it also actually gets into northeastern Russia and Kamchatka Island. So it has this really big geographic range. And within that geographic range, it has an incredible ecological amplitude. So that means that it's found in all kinds of different habitats. So this is an example of a species that is found at very high elevations. So this is Castellais nana, the Lapidicula form, and it's situated in subalpine to alpine environments. We find it in the Sierra Nevada and east into the mountains of Nevada. And here, this population that you see is these pictures were taken in the White Mountains. If you look in those landscape pictures on the right and in the center, you can see that, you know, the tiny plants in the foreground, but if you look kind of along the horizon, you'll see a bit of a purple or a dark brown coloration to the rocks. Those are all Castellaiana. Like, this was a bumper crop this year when I took, well, this is a couple of years ago when this picture was taken. They were extremely happy up there. And the picture there on the left is, of course, the close-up of this particular species. So Castellia can be found in these really high alpine environments, but they can also be found in very low elevation areas near the coast. So this is a picture of Castellia latifolia. And this little guy is found in coastal sand dunes and sandy bluffs, and it's endemic here to Central California. So here we got the habit on the left. It has that deep purple herbage, so all of that bright green is a different species. There in the center, we can see a close-up on its inflorescence. And there on the right, I have a picture of the leaf. So this species has a very kind of thick and waxy, almost kind of succulent leaf. And it snapped really simply and easily when I was fiddling with it. So high elevation, very low elevation. Castellia can also be found in really wet environments. So this is the species Castellais cousciai. This species is widespread across the meadows of the Intermountain West. And here, this picture is taken in central Idaho. So this is a species that really likes to have its feet wet. Yeah, just really enjoys these kind of, this is a moderate elevation meadow here. And Castellia can also be found in really dry areas. So this is a picture of Castellaius fabrita. And this species is found in the high deserts of Utah and Western Colorado. And I took these pictures when I was at Canyonlands National Park. So that's in east central Utah. And this is a group that is growing very closely, tightly associated with sandstone in these really dry, arid environments. So high elevation, low elevation, wet areas, dry areas. It can even be found in more extreme environments than some of the ones I've mentioned. And a great example of that is Castellaius genosa. So this is a group this is a species that is found in these really harsh alkaline soils, really basic soils that are in these hot spring areas in the low valleys of Nevada. And you can see the close-up picture of this really strikingly beautiful plant on the left. And if you kind of squint on the right, you can see it hanging out at the base of that really big plant that's there in the middle. They can be pretty cryptic. They blend in really closely with the surrounding environment. When we were out at this site trying to find this plant, I almost stepped on the first one and I kind of screamed a little bit when I saw it. This one's a very rare and a very endangered species. And in fact, it's only known from two populations, one of which is likely extirpated. But anyway, the point of this slide is to show that this species can tolerate very harsh and extreme environments. Okay, so geographically and ecologically, Castellia is found almost everywhere. It's very, very widespread, and it can be found in almost any kind of habitat. So the next set of diagnostic traits that we use to identify species, or we try to use to identify species, have to do with its morphology and what the plant looks like. So many of the species have what we call overlapping floral morphology. So morphology that doesn't kind of distinguish a single species, but can actually be assigned to many species at once. So quickly, I just want to show you kind of the general nature of morphology in Castellia. So this is kind of a diagram of just a basic plant. This is the habit, the entire plant. If you take off one of the tops of one of those branches, that's where the inflorescence is, where we have many flowers. So that's what this looks like. And if we picked off just one of those flowers and we dissected it into its 3 major pieces, we would find the bract. This is the leaf just below each flower. The calyx, this is also called the sepals, and the corolla, also called the petals. And usually it's characteristics of these last 4 elements that we use to define species. So the shape and the lobing of the bracts is usually diagnostic. The depth of the sinuses between the calyx lobes, and this gets really confusing because these are tubular flowers and some of these lobes are all the same height and it's really hard to know which is which. So the depth and the shape of the depth of the sinuses and the shapes of these lobes. And then the length of the beak and the characteristics of the lower lip are oftentimes the floral features that we kind of cue in on. And we also use characteristics associated with the surface, things like the pubescence or the hair that is on the plant and whether it's glandular. And we also use inflorescence colors to try to diagnose species. So this is the basic kind of suite of characters that we try to find in all species of Castellia. But there are some kind of major morphogroups or kind of forms within, that kind of fall within these major pieces of the flowers. So this slide shows the major floral forms of the calyx and the corolla for that represent all the species of Castilleja. So the different form is organized here by color. Within each color, you have the calyx on the left. The corolla on the left and the calyx on the right. And then underneath that, there's a little box that has the name of the group or the morpho group that we've assigned, how many sections and how many species are within each of these major morpho types. Now, 4 of these, you'll notice, are quite different looking than everybody else, particularly those first 3. And they also have very few species. In fact, only a single species assigned to each of these major morpho groups. So we don't really kind of think of them as being representative of the major morphologies of Castilleja. And in fact, those first 3 over there, we actually thought they were different genera for a long time. So very distantly related to Castilleja. And it wasn't until we started building phylogenies that we understood that they were actually part of Castilleja and they just had really different floral shapes. But anyway, the major kind of 5 groups that we cue in on and that we see the most of are represented here. And so you'll see that a number of different species have been described within each of these major floral forms. And unfortunately, once you're inside one of these major floral forms, it's really hard to tell the different species apart. On top of that, other characteristics that we might use that have to do with the floral features, other characteristics that we might use to try to identify different species are poorly understood or are not as informative as we would like to believe they are. So for example, characteristics that have to do with pollination are important for gene flow and for understanding how species can breed with one another. And frankly, we don't have a ton of understanding about the details of pollination in this group. So for example, we think that these kind of short-beaked forms are insect pollinated. And we think that these longer-beaked forms are hummingbird pollinated. But there have been very little formal tests to see if that's actually the case. And we'll see in a couple of slides some good evidence that these very different floral morphologies actually share pollinators, which is another thing that makes distinguishing these species so challenging. So we don't know a ton about their actual pollinators. And another character that we cue in on is the color of these flowers and the color of their inflorescences. oftentimes those help us understand something about the pollination biology. But Castilleja is a bit tricky with their colors. So we would love to say that, species X is always red and species Y is always white. But we see many different cases where these colors seem to be much more variable. And here I have some really nice pictures that illustrate that. So, the picture from my opening slide, this kind of rainbow of pictures here. This is Castellaius miniata from British Columbia. And normally this is a red species, or we think of it as a red species, but this population had individuals that ranged from white all the way to red and every color in between. Another species that I've seen evidence of this is Castellaius pylosa. This is a collection of individuals taken within a very, very large population that did span some elevational difference, but still a cohesive population that ranged from creamy green yellow all the way to a dusty kind of pink color. And then these images on the right side are some specimens that we just mounted in the herbarium at Cass. And these were a set of beautiful collections. A woman was in Bodie Hills, east of the Sierra, and she recognized that this population had multiple colors of individuals. And so she made separate collections of this, which was really, really fantastic. So this is another example about how we want color to help us tell where species differ, but we can oftentimes get confused by color because there's so much variation within a species sometimes. So another kind of biological aspect of Castellaius that very likely influences its evolution, but that we still don't fully understand, is the role of parasitism in this group. So Castellaius is actually a parasitic plant. And some people are very surprised to hear that. I was certainly surprised the first time. I always think of parasites as being something that's nasty and creepy. But really, all that a parasite is an organism that can create or a plant that can create a hostorium. So this is a special organ that the roots of Castellaius will create when they encounter the roots of a putative host or a potential host species. So here at the bottom left of the screen, we have a really nice close-up photo of what this looks like. So the host root is that darker root that we see going from kind of at a diagonal. And then the Castellaius root is coming from kind of lower down, and it has met the host root and created the hostorium. So this is that organ. Now, if we look kind of at a cellular level at what's happening at the hostorium, we'll see something similar to what we see in that center picture. So this actually isn't Castellaius. This is a very close relative called Striga, but it also forms this hostorial connection. And this is a beautiful kind of histological section of this hostorium. And Castellaius hostoria probably looks a lot like this. So the tissue at the bottom of that cellular diagram, that's the host root. And the hostorium is kind of there at the top of the frame. And you can see that it is pushing its cells into the vascular tissue of that host root. It's actually punching a hole into the root tissue and accessing the vascular highway, which is basically what the host plant uses to move water and sugar around its body. It's punching a hole in there and it's gaining access to it. So the parasite actually kind of slurps up water and sugars that are in the host itself. So Castellaius is what we call a hemiparasite, which means that it can form these hostoria, it can make these parasitic connections, but it can also still perform photosynthesis. So it can behave as a regular kind of plant. And in fact, what we call these are facultative hemiparasites, which means that they can kind of take it or leave it, whether they form a parasitic connection or not. So this picture that I just flashed up is Castellaius fabrita. We saw him in one of our earlier photos. And here it looks like he's growing completely out of the rock with no apparent host in sight. And so this is a good example of this individual completely able to complete its life cycle without having a host. But we do know that they are parasitic. They will form parasite and host connections whenever they can. And trying to understand who the hosts are and what their relationship between Castellais and its host is still a big open-ended question. So this last picture that I popped up is a picture of an intact connection between Castilleja there on the left and its host there on the right. And this was a recently described species from Colombia. And, you know, the fact that they were able to remove this plant from the ground without destroying those hostorial connections is pretty impressive. You know, I've tried many times to dig plants up without snapping those really delicate connections, and it's really quite hard. So it's hard to know exactly who Castilleja is connected to. So that's kind of an open-ended question for the biology of Castilleja. Okay, so the last kind of suite of diagnostic traits that I wanted to talk about with respect to Castilleja has to do with its genome and the genetic information that we can use to try to understand species boundaries. One thing, the longer that I work in Castilleja, the more I realize how complex the genetics of Castilleja are. And it's really made it very challenging to try to access the information that the genome holds. And there's really kind of three major reasons why the genome is so complicated and why collecting genetic data from Castilleja is so difficult. So first of all, they are an extremely young lineage. So this doesn't sound really young, but Castilleja is probably, the whole genus itself is probably between 4 and 6 million years old. So that's a long time from our perspective, but from an evolutionary perspective, it's a drop in the bucket. It is an extremely short amount of time. On top of that, most of the species diversity that we know of or that we've described at this point in Castilleja was probably generated in the last several 100,000 years. So an even younger group of species than ever. Like it's just a very young group. And for that reason, a lot of the genetic characteristics and traits between species haven't completely sorted out yet. They're very overlapping and they can be very confusing. On top of that, many species of Castilleja have duplicated their genomes. So this is a thing that we call polyploidy in plants. Lots of different plants do this to some degree, and the more we look for it, I think the more that we find it. But Castilleja has been kind of doing this like a champ. Many different species are polyploids, and what's more, within a species, there's oftentimes varying levels of polyploidy. So some species we've found individuals that have 4 copies of their genome, some that have 10 copies of their genome, some that have 20 copies of their genome. It seems to be a kind of a path that many different populations of species have taken. And of course, the more copies of your genome you have, the more complicated any kind of genomic analysis becomes. The last thing that makes the genetics of Castilleja so challenging is the fact that we see a lot of gene flow that happens across species on a pretty regular basis. And this can often happen across what we think of as very distantly related species. So here I have a visual example of this. This is from the White Mountains. There on the left, we have one of our, quote, hummingbird pollinated species. And then at that same location in the same area, we have another species that's supposedly pollinated by insects. And at that same site, we see hybrids between the two. So that picture on the right has a beautiful blend of characteristics of both of the two potential parents. And this is what we call just a hybrid zone, where these two species that are very different morphologically co-occur. But clearly they're exchanging genes if there are hybrids in the area. And in order for them to exchange genes, they have to exchange pollinators. So this gets back to the idea that our understanding of pollination in the group is limited, and it really hampers our ability to understand how gene flow happens too. So these are the three main reasons why genetics in the group have been really hard. But during my PhD, I attempted, and I am still attempting, to sort out all of these different complications, because the genome is really a place that harbors some very unique and important information to understand evolution in the group. And so I wanted to share one big phylogeny with you to show you some of that work. It is a big tree, and I'm sorry for the size of it, but I was trying to get enough of it on the screen to show you. But here I have a phylogeny on the left that has over 500 tips. So this is 500 different individuals that have been sampled. I've sampled their genomes. And here I was looking specifically at their chloroplast genomes. And I used a phylogenetic method to build this diagram about how these individuals are related based on their chloroplast genome. And then what I did is I color coded the tip to correspond with where they were found in geography, in geographic space. And so that top group on the tree has red tips. And if you look over at the map, and again, I'm so sorry for the size of the map, all of those red tips are found kind of in the northwestern part of the United States, up into Canada and over into Alaska. So all of those individuals in that area, according to our phylogeny, are most closely related. We see similar groupings in the other major parts of the tree that correspond with the dark blue colors, the green, the orange, the yellow, etc. They're all from these different geographic regions. Now, the crazy thing is that some of our most widespread species that exist across the whole length of North America and into Mexico are all found spread entirely across the tree. So they pop up in all of these different geographic clades because they're distributed broadly geographically. But if we think about what a species means, we usually assume that all members of a species will be most closely related to one another. We would expect that they would be in the same clade and not actually distributed across these geographic clades. So this is a real perplexing kind of signal that we see when we look at the genetic data of Castellia. So a lot of my kind of current research is trying to sort this kind of stuff out. Okay, so the last few slides that I want to show are just some kind of examples about what this challenging taxonomy looks like from the perspective of a person in the field looking at these plants. Yeah, I've got 5 different species complexes to introduce you to. And each of these complexes are places where I've already gathered some types of data to help me distinguish where species are, and where I continue to gather other types of data to try to understand species boundaries. So the first one is one of my favorites. This is the Castellia pylosa species complex. And this is a number of species that we've already seen from some of our other slides. But these are individual species that, when we look at them kind of one by one, they might look very different from one another, with the exception, of course, of that first row. Those all look really similar to me. But particularly the ones on the bottom row, they look really similar, or they look very distinct. And so they've been named different things. But the problem is when these species co-occur, they tend to morph into variations that look very similar to one another, excuse me. So some of these are high elevation species and others are moderate elevation species. And so when those high elevation species like Nana lapidicola in the top right there, when it starts reaching its lower elevational limits, and Castellia pylosa there on the left starts reaching its higher elevational limits, they begin to co-occur and they begin to look exactly alike. And so it's really hard to distinguish them. And then these 4 species there at the bottom are kind of distributed on these isolated mountain peaks. But genetically, they look exactly the same as pylosa and Nana. And so this group is like this kind of sort of morphologically distinct, sort of not morphologically distinct, genetically confusing group of species. So this is one complex that I work with. A similar one is the Castellais cousciai complex. These are those wet meadow species from the Northwest. They range into the area of Castilleja pulchella, which is in the Central Rockies. And again, when these different species reach their elevational or habitat boundaries and edges, they tend to look more and more similar to one another. Castilleja ambigua is one that's really close to us here on the Pacific coast. The nominant form, Castilleja ambigua, variety ambigua, extends all along the coast of North America. And then it has these pockets with these morphologies that are very different and unique. So variety and salutata there in the center is around San Luis Obispo County, and Humboldtiensis there on the right is found up in Humboldt County. This widespread species, or the widespread variety ambigua, also has a lot of morphological variation within it. And there are places where variety ambigua actually looks a lot like some of the other named varieties. So for example, that small inset photo is from a plant found at Tamales Bay that looks a ton like variety and salutata, which is found in San Luis Obispo. So the question is variety and salutata more widespread? Or is variety and salutata kind of a misnomer? And it actually is just some of the variation that exists within the widespread variety ambigua. Another complex that is very commonly found in Western North America from the Southwest all the way up into the Yukon is the miniata complex. So all of these pictures here are from a group of, are all named varieties of the widespread species miniata. And this is another one that has been remarked on formally in the literature about its challenging, its difficulty defining or to identify species here. So in the Intermountain Flora, in volume 4, they say Castilleja miniata is one of the most widespread members of the genus, and the morphological complexity is exceeded by few. Throughout the Intermountain region, Castilleja miniata remains distinctive, but beyond our borders, these distinctions often break down. That could be because of hybridization with species that exist there. It could be because of unique habitats at the edges of its range. These are things we don't know. Okay, the last species complex is the Castilleja, well, what I'm calling the coastal California Castilleja complex. I swear I mix those words up every single time I say it, a lot of C's. This is composed of primarily of Castilleja affinis there on the left, latifolia in the center, and whitei on the right. And then there are these other 2 species that I don't have pictures of here, Mendecinensis and littoralis. And I'm also pretty sure that it includes these other 2 members that are very common for us here in the Bay Area, subinclusa there on the top right, and foliolosa on the bottom right. And this is a group of plants that, again, at first glance, they look pretty different from one another. But when we get into areas where these species overlap with one another, there are a lot of intermediate forms. So there's probably some hybridization happening across these species. And on top of that, these species switch colors. And I'm sorry I don't have visual examples of this, but whitei, that yellow one in the center, It's only yellow kind of in our neck of the woods. Everywhere else it's red. And foliolosa in the bottom right is primarily reddish orange until you get down to Monterey, where apparently it turns yellow. And so, you know, what's driving those variations and where species boundaries are when things are so different across ranges is one of the questions that I have in this group. This is the complex that I've just recently started working with, and I'm pretty excited about potential here. So anyway, I hope that these complexes have given you a sense of some of the challenges of knowing exactly how many species we have in Castilleja and what species mean in this group. Ultimately, these complexes are places where we need to perform that careful species delimitation in order to understand where the boundaries between these species are. And as I mentioned in the beginning, you know, distinguishing species is the first step. We need to know what these units are that we're studying in order to put them back on the phylogeny to try to understand the processes that are generating it. And ultimately, once we have that knowledge of where the species are, we have the phylogeny, we can describe the steps of evolution, then we can start to feed everything back into, you know, that whole kind of cyclical and dynamic kind of work that is systematics. So yeah, so I feel like I threw a lot of this at you, and but I hope you have a sense of kind of the work that I do. So with that, I will say thanks very much for the invitation and for your attention this evening. I'm super happy to take questions, any kind of questions. So
nice. Fantastic, Sarah. That was really, really fascinating stuff. And I'm never going to complain about Impedinax flycatchers again.
Okay. I appreciate that.
Yeah, that's kind of mind-blowing, the color changes.
Yeah.
All this time we thought we might be getting it to species. Now I'm like questioning all of that, what we were calling these
Welcome
things.
to my world, Eddie. Welcome to my world. Yeah. Actually, when I first contacted the person who would become my advisor in graduate school, you know, I contacted him and I was like, Dave, I want to do a PhD, and I know you. I'd really like to work with you. I want to study under you. And he said, great. I'm working in Castilla. And I was like, oh no, I'm scared. Like I've never successfully identified a Castilla. I'm intimidated by this group, you know. And he kind of laughed and he said, oh, you'll learn, and you know, you'll figure it out. And he was right. I mean, there's still so much to know, and there's still so much that I don't know, but
yeah,
I'm trying. So
mind-boggling how they can change colors like that.
I know. It's incredible.
Yeah, neat. Well, there are some really good questions in here. If you've got some more time to share with us.
Absolutely.
Or as asking, do the different morphological differences in Castilleja all have known functional significance?
That's a good question, and I would say no. We don't necessarily know the functional significance of these morphologies. You know, we very quickly tie them to pollinators, since pollinators are what's moving pollen from one flower to the next. That's what's moving genes around. And that movement of genetic material is part of what contributes to species and contributes to the success of the organism, the individual, right? So that it can be pollinated, it can make seeds, it can get its gametes into the next generation. So we oftentimes go towards morphology or towards pollination as a way to understand the function of a morphology. That said, one thing I didn't mention here, which some might say could have a functional role, Castellaius flowers are heavily, are oftentimes for lack of a better word, infested with all kinds of eggs and larvae of other insects. So I think that they're a common breeding ground and rearing place for lots of different types of insects. In fact, when I put Castilleja in my press, if I don't dry them very quickly and very hot, well, not super hot, but pretty hot, those insects will start eating their way out of the flowers. And my inflorescences, when I pull them out of the press, they've all kind of disintegrated. So anyway, yeah, I think that there are some other functional aspects of the morphology that we haven't really considered and haven't formally documented either. So.
Cool. Well, that's a nice lead into to Henry's questions. How do scientists formally test whose plants are pollinated by?
That's a really great question. So a lot of that is done by more explicit experimental work. So, you know, we can sometimes pick up the signatures of parents from genetic data, but you usually have to have really high resolution genetic data. You have to have really precise, detailed information from all of the potential parents. And very rarely do we have that. And it's really only in model systems like Arabidopsis, which is a model system in plants, where we have that kind of scale of information. So usually we fall back on experimental procedures where we are either hand pollinating ourselves, literally using a paintbrush, a literal paintbrush, to move pollen from one flower to another. And then you can kind of exclude those flowers from any future pollination by putting bags on them so that insects can't access them. And so you control who's pollinating then. And we do experiments that way. Sometimes you can capture the pollinators themselves and take the pollen from the pollinator and analyze it and identify, if not an individual, or at least a species that pollen comes from. I've also heard about people following pollinators around, but some of the problems there happen, you know, oftentimes the pollen that gets presented to the flower is a ton of pollen, and it can come from any number of different plants. And on top of that, when the pollen touches the stigma, there's a whole lot that still has to happen before pollination actually happens, before the sperm is actually delivered to the egg. And so, yeah, these are usually like really big kind of experimental endeavors for us to really clearly understand who's pollinating what, exactly where pollen came from, things like that. Yeah.
Cool.
There's a question, are plastids inherited maternally in plants? And does that matter in terms of understanding lineage?
That's a great question. Yes, that's absolutely correct. So plants have actually 3 genomes. They have the nuclear genome, which is biparentally inherited. So one copy comes from the mom and one comes from the dad. And then they have these other 2 organellar genomes, the chloroplast genome that is associated with photosynthesis. And then they also have a mitochondrial genome that is, well, it's not as heavily used in plants the way that it is in animals, but they do have a mitochondrial genome. But both of those genomes are, for the most part, uniparentally inherited and maternally inherited. And yeah, so when we build a phylogeny with one of those uniparentally inherited genomes, then essentially what we're seeing is just kind of one side of the story. We're basically picking up the signal from one parent about how things are related. Oftentimes when we engage in like genetic research, we oftentimes go for the easiest genome to get information from to begin with. And in chloroplast or in plants, they have a bazillion chloroplasts, like stupid amounts of chloroplasts. So it's really, really easy to get the genetic material from them and to build big databases. And so that's almost always where we get our first kind of hint at the genome. In Castilleja, it was also really important to start with the chloroplast because, as I mentioned, the nuclear genome is oftentimes duplicated many times over in Castellaius species. So they're polyploids. And we don't think that the chloroplast genome is affected in that same way. So while we're only getting half of the story, it's much easier because it's easier to access that information and we don't have to sort out all of these different copies. But nevertheless, the nuclear genome is where we're going to find, I think, our most important answers. So, yeah.
Cool.
There's a question about that yellow coastal plant.
Mm-hmm.
Could that be an ecosystem with a different plant palette that's just that is a
Parasitic on it? Yeah, so could the parasite of since it parasites on certain host plants, could that impact the color?
That's a great question, and I don't know the answer to that. When we think about what is being shared across that hostorium, we usually just think about it being water and sugars. So Castellaius is really just trying to get a cheap meal
Mm-hmm.
as much as it can. But there is possibility that other compounds from the host plant are being pulled across that barrier as well. So there have been some studies that have shown that Castilleja that is connected to a legume or to members of the Asteraceae, the sunflower family, oftentimes will have some of the chemical warfare that those plants have embedded in the tissues of Castilleja. So there can be a transmissal of or transmission of other types of molecules. Now, if those molecules can actually affect the flower color, I don't know. It's a good question, but my suspicion would be that it would not be the case. Usually flower color is encoded for. It's something that is created by the plant. So it needs to be coded for as a protein in the genome. And it won't be as susceptible. At the same time, there are examples of plants that do react to their environment, like hydrangeas, for example. If you put them in a different pH soil, the flowers will change colors.
Right.
So yeah, no, it's a good hypothesis. It'd be a great one to test.
Cool. And then kind of along those same lines, Susan's asking, are there certain host plants or plants that the Castilleja excerpta subspecies latifolia targets, as to be a parasite of?
Yeah. I don't know for sure about variety latifolia. And I don't know for sure about Castilleja excerpta either. I do think that, when these species are described, their close associates are typically, identified. And I think we like to hypothesize that we know who it's growing on or connected to because of proximity of the stems. But it's really hard to know what's happening underground. And
Right.
The connections between the roots of Castilleja and its host are very delicate. And I've never successfully dug a plant up without destroying those connections. What's more, those roots can travel pretty far. So it very well could be connected to the thing that it's most close to. I mean, that's a very logical assumption to make. But we don't know if Castilleja will connect to multiple species at once. So maybe it's connected to that nice coyote bush that it's sitting in between the branches of, but maybe it's also kind of hanging out with the grasses that are a meter away. and it's just been growing its roots slowly towards the grasses to steal some of their water and sugars too, or to take advantage of some compounds that are in the tissues of those plants. That's a possibility as well. You know, I think that some people have tried to standardize how we guess what they're connected to, but until you actually dig them up and look for those connections, I think it's really hard to know.
Right, right. So do you keep track of which insects are pollinating which, I mean, is there, because that's something like we're always really curious about, you know, because of the whole biodiversity issue and the loss of biodiversity. We're like, okay, which plants, you know, are best supporting, you know, its neighbors.
Right. Yeah. Yeah. I try to do that to the best of my ability. I am woefully underskilled at identifying different types of pollinators, but I am learning. The work that I did during my PhD, I was trying to cover and to sample as many species as possible in 2 field seasons. And so I was driving, I think at one point I drove like 15,000 miles one summer, you know, from Southern California up to Alaska and back. Like I covered so much ground. And so anytime I, you know, encountered one of my species or one of my collecting populations, I had to leave basically pretty quickly. So I never got to stick around to look for the pollinators. But now that I'm here and now that I'm around the bulk of the diversity of Castellia here in California and in this floristic province, there's going to be so much more opportunity to spend time at populations to do a better and more thorough job documenting them. So the short answer is yes, absolutely. I do what I can.
Cool. Well, do you work with entomologists then sometimes to?
Yeah, exactly. And that's going to be the fastest way for me to gather that kind of data, is taking out experts who know more than I do, and then certainly that I can glean information from too. So
nice.
Yeah.
So one last question, if you don't mind, from Bob. Hummingbirds tend to pollinate red plants. When Castilleja changes from red to white, do the white forms get as much hummingbird action?
That's a great question. And I don't know. I hate that all of my answers are, I don't know. So I have seen, you know, I've seen, you know, the long tubular flower form we attribute mostly to hummingbirds. We have seen hawk moths at those tubular flowers too. And I have witnessed the hummingbirds coming to that floral morphology. And I have, of course, seen hummingbirds at non-red flowers as well. So my guess is that those pollinators still manage to get to those white flowers if they have the right shaped flower. I think shape matters quite a lot. And it could also be, and I think this is very likely too, that each species is probably pollinated by many different things. And so even if the hummingbird is no longer happy about the fact that the flower is white and it goes somewhere else, you know, there's probably another insect that's there, or another insect that would probably do the job as well. So.
Well, this has really been fun, a real brain tickle here.
Oh, good.
We so appreciate you joining us and sharing your research.
Yeah, it was a pleasure to be here. Thanks for listening to me talk so long. I think I said I would keep it at 40 minutes and then I talked almost an hour.
No, this has been a blast. We loved it. Yeah.
Oh, good.
And hopefully we'll get to see you in person here sometime soon.
Yeah.
We'll go birding.
Yes, I would love birding. I would love to do that. And maybe I'll see you guys out botanizing too.
Yeah. Well, thanks for being here, and thanks to everybody else. And we'll look forward to connecting with you all soon.
All right. Thanks again.
Bye-bye.
Bye.